The overall goal of the following experiment is to use the fluorescent proteins morange2 and Dronpa for dual probe optical highlighting to selectively label 4 individual cell populations or cellular compartments. This is achieved by first illuminating cells with low-intensity 488 nanometer excitation light to switch off all the green Dronpa fluorescence. As a second step, cells are exposed to high-intensity 488 nanometer excitation light, which photoconverts the orange fluorescence to red. Next, cells are exposed to 800 nanometer 2-photon excitation in order to switch on Dronpa fluorescence again. Results are obtained that show it's possible to label cells in 4 distinct combinations based on dual probe optical highlighting with morange2 and Dronpa.
The main advantage of this procedure over existing experiments like subcellular fractionation is that it's done on live cells. This procedure can help answer key questions in beta cell biology, such as why only a small fraction of insulin vesicles are secreted. Although this procedure can provide insight into beta cell function, it can also be applied to other systems, for example, tracking cancer cells in vivo.
A fluorescent protein droplet sample consists of an octanol water emulsion with the fluorescent protein residing in the water phase. This emulsion is sandwiched between a microscope slide and a 22 mm square cover glass for microscopy applications. At the beginning of this procedure, the microscope slides and cover glasses must be cleaned and coated with a hydrophobic agent. Clean the glassware by washing it for 5 minutes with acetone. After washing, leave the glassware to dry by air.